The overall goal of this transduction protocol is to stably introduce reporter genes in patient-derived xenografts, or PDXs, to facilitate their use in experimental and spontaneous metastases models.
This method can help answer key questions in cancer research, such as the extent of spontaneous metastasis from patient-derived xenografts. The main advantage of this technique is that labeled PDX can be tracked using in vivo imaging, facilitating the assessment of organ-specific metastases without the extensive histological analysis post-mortem. Demonstrating this protocol will be Jessica Finlay Schultz, a postdoc who routinely applies this protocol for the label of breast cancer patient-derived xenografts.
Using aseptic technique, begin by using a scalpel to cut the skin around the tumor. Then, pull the skin with forceps and use a clean scalpel to separate the skin from the tumor. When the tumor is completely exposed, use a clean set of forceps and scalpel to transfer the tissue into 5 ml of wash medium on ice. Next, discard the medium and wash the tumor 2 times with 10 ml of HBSS supplemented with HEPEs. Place the rinsed tumor in a sterile pre-weighed 60 cm tissue culture plate, and weigh the plate to estimate the tumor mass. Using a scalpel, cut the tumor in half. Then cut a 3 mm disc from one of the halves and place the disc in 10% formalin for 24 hours. Add 200 microliters of HBSS with HEPEs to the rest of the tumor, and use forceps and a clean scalpel to mince the tissue into the smallest possible pieces. Transfer the fragments into a sterile 50 ml conical tube, and add at least 1 ml of digestion buffer supplemented with antimycotics and antibiotics per 100 mg of tumor. After 3 hours at 30 degrees Celsius and 125 to 200 rotations per minute, add 35 ml of wash medium to stop the digestion and filter the resulting tissue slurry through a 70-micron nylon mesh into a new 50 ml conical tube to remove any undigested tissue. Collect the cells by centrifugation and resuspend the pellet in 1 ml of red blood cell lysis buffer for 5 minutes at room temperature. At the end of the lysis, restore the tonicity with 10 ml of wash buffer and strain the cells through a 40-micron filter to remove any clumps. Centrifuge the cells again and resuspend the pellet in 5 ml of wash buffer on ice. Then count the number of viable cells by trypan blue exclusion. To enrich for the human epithelial cancer cells using lineage plus mouse cell depletion, transfer up to 1 × 10⁷ cells into a new 5 ml polypropylene tube, and add 2 ml of epithelial enrichment buffer to the cells. Collect the cells by centrifugation and use a pipette to completely remove the supernatant. Next, resuspend the pellet in 40 microliters of ice-cold epithelial enrichment buffer, and add 10 microliters of biotin antibody cocktail per 1 × 10⁷ cells. Mix by gentle pipetting and incubate the cell solution for 10 minutes at 4 degrees Celsius. At the end of the incubation, mix the cells with 30 microliters of epithelial enrichment buffer per 1 × 10⁷ cells, followed by the addition of 20 microliters of antibiotin microbeads with gentle mixing. After 15 minutes at 4 degrees Celsius, wash the cells in 3 ml of cold epithelial enrichment buffer and carefully aspirate the supernatant. Resuspend the pellet in 500 microliters of epithelial enrichment buffer on ice, and place a column in a magnetic separator. Rinse the column with 0.5 ml of epithelial enrichment buffer, and place a new polypropylene collection tube under the column. Then slowly add the labeled cells over the column. collecting the enriched, unlabeled, lineage-negative, tumor cell fraction effluent, followed by 3-column rinses with 500 microliters of fresh epithelial enrichment buffer. To transduce the tumor cells, centrifuge the isolated cells for resuspension in 2 milliliters of Mammosphere medium. After counting, centrifuge the cells again and resuspend the pellet in 2 milliliters of Mammosphere medium, supplemented with 20 microliters of polyborine. Next, plate 2 times 10 to the 5th viable tumor cells per well in an ultra-low adherence 6-well tissue culture plate, and add lentiviral particles to the cells at a multiplicity of infection of 10. Swirl the plate to mix the virus with the cells, and incubate the co-cultures at 37 degrees Celsius and 5% carbon dioxide for up to 96 hours, adding 500 microliters of fresh Mammosphere medium after the first 24 hours. When at least 10% of the cells are GFP positive, transfer the transduced cells from at least one well into a 15 milliliter conical tube on ice, and wash the well with 1 milliliter of Mammosphere medium. Pool the wash with the cells and centrifuge the tumor cells in 10 milliliters of HBSS plus HEPES. Finally, resuspend the cells in 50 microliters of basement matrix extract on ice, and load the embedded cells into a 0.5 milliliter insulin syringe for reimplantation. Some high-titer viral vectors express a fluorescent marker that allows estimation of the transduction efficiency in vitro as early as 24 hours after infection. For most PDXs, the GFP expression will be delayed up to 72 hours after infection, at which point the formation of cell aggregates is commonly observed. After extracellular matrix suspension and reimplantation, the labeled tumor cells regenerate tumors that can be tracked using bioluminescence or fluorescence. A successfully labeled PDX will be nearly 100% GFP positive at the first generation post-transduction, and will remain so in subsequent passages. However, some PDXs will exhibit a patchy distribution of GFP positive cells, indicating a suboptimal in vitro transduction. Immunohistochemical tumor tissue staining reveals that the expression of critical markers, such as epidermal growth factor receptor and pan-cytokeratins, are conserved even after several passages and reimplantations. Experimental models of metastasis provide an alternative to studying organ tropism and organ colonization steps in the metastatic cascade. Indeed, a transduced PDX injected at 2.5 times 10 to the 5th cells per mouse forms metastases in the liver that are easily identified with luciferase imaging in vivo, and in the lungs that can be visualized by fluorescence microscopy ex vivo.
Once mastered, the dissociation and labeling of these PDXs takes about 4 hours. The actual expansion of the tumor can take 8 to 12 weeks, depending on the tumor and system used. When attempting this procedure, it's important to use a high-titer virus with a reporter gene so you can monitor both transfection efficiency and tumor growth in vivo. Following this procedure, other methods such as in vivo metastasis studies can be performed to answer other questions on tumor progression, metastasis, and response to treatments. This technique paves the way for breast cancer researchers to explore multiple aspects of tumor progression using novel PDX models that better represent tumor heterogeneity. After watching this video, you should have a good understanding of how to label dissociated tumor cells with high-titer lentiviral particles, which will allow you to both trace tumor cells in vivo as well as potentially manipulate the expression of genes of interest. Don't forget that working with lentiviral particles can be extremely hazardous, and you should always take proper precautions when performing this procedure.